Now, believe it or not. We good? Okay. So today we're going to finish our discussion of chapter 22, finishing off the hydrolysis of amides. Then we'll discuss our biological carboxylic acid derivatives that are used in reactions in biochemistry. We'll conclude with the discussion of the chemistry of nitriles, our compounds that are not carboxylic acid derivatives, but do behave somewhat similar. Another announcement we have is that there is a new Bacon quiz that is available starting today on the chemistry of carboxylic acid derivatives. So you can feel free to use that as part of your preparations for exam 2, or you can save it and use it for the final exam. At the end of class on Wednesday, we went over the acid-promoted hydrolysis of amides. We said that the mechanism was the same as the Fischer esterification until you got to the last step, and we used the amine as the base to make an ammonium ion as opposed to using the conjugate base of our acid. I've got the mechanism for basic ester hydrolysis on the board here. And as you might imagine, it has parallels to the saponification reaction, or the base-promoted ester hydrolysis, but there is one key difference. Starts out the same. Our hydroxide is going to attack the carbonyl carbon, generating a tetrahedral intermediate. Whenever we form a tetrahedral intermediate, we want to ask ourselves, what is the best leaving group attached to the carbon? Because that's what's going to depart when we have our elimination. In this case, it's the OH group that just arrived. And so probably 999 times out of 1,000, what's going to happen is this tetrahedral intermediate will collapse to eliminate hydroxide and just go back to the starting material. But it's not impossible, and every once in a while, maybe once in 1,000 or once in 10,000 times, you're actually going to eliminate out NH2 minus, okay? So that's going to be a very unfavorable equilibrium to do that, as I've indicated by these arrows here. But once we form these two entities, the carboxylic acid and NH2 minus, they are going to react very rapidly and irreversibly in an acid-base reaction. This is a very strong base, and so it will irreversibly deprotonate our carboxylic acid, giving us a carboxylate and producing ammonia. Usually to determine whether or not an acid-base reaction is thermodynamically favorable, we look at the pKa's of the acid and the conjugate acid. In this case, the acid is a carboxylic acid. The pKa is typically between 4 and 5. The conjugate acid in this case is ammonia. The pKa of ammonia is around 33, if I remember correctly. Okay, but it's above 30. So for this reaction, you would have an equilibrium constant of about 10 to the 25th. So you can see why we've only drawn one arrow. We haven't drawn the reversible equilibrium arrows that we would frequently draw. So this third step of the base-promoted hydrolysis of an amide is irreversible. So even though we only produce a tiny amount of the carboxylic acid and the NH2 minus, that tiny amount of those two components will react rapidly and irreversibly to generate the carboxylate ion, the conjugate base of our carboxylic acid, and NH3 minus, or NH3 ammonia, sorry. So this is another way of driving a reaction through an unfavorable equilibrium. Earlier on in this section, we've used Le Chatelier's principle to drive reactions through unfavorable equilibriums by removing a product. In some cases, we've used large excesses of reactants. Here, what we're doing is having an irreversible step after the unfavorable equilibrium. And so in a sense, we're removing products because we're removing the products of the unfavorable equilibrium by this irreversible reaction. And so according to Le Chatelier's principle, this tetrahedral intermediate will react to form more of this as those components are consumed. Are there questions? Yes, Grace. So we do use hydroxide in this case. And of course, if we were hydrolyzing an ester, it would be an alkoxide at this point that would react. Okay? But the reason we have NH2 minus here is just a natural consequence of the fact that we were hydrolyzing an amide. Okay, so we're locked into having NH2 minus or another nitrogen base. All right, if this were a secondary amide with an R group on the nitrogen, then it would be NHR at this point, but it would be a negatively charged nitrogen. Okay, so we have to use a negatively charged nitrogen here because that's what is eliminated when you're hydrolyzing an amide. And then we do use hydroxide at the beginning. Other questions? All right, so I have up on the screen the structure of penicillin. So amides are our most stable carboxylic acid derivatives. And these reactions, these amide hydrolyses, usually have to be heated in order to have them occur on a reasonable time scale because the amides are the least reactive of our carboxylic acid derivatives. But penicillin is special. It has two different amides in it. Penicillin is actually not one molecule. It's a collection of related molecules that have different side chains here on this amide, this acyclic amide. But we have a second amide of penicillin that's embedded within this four-membered ring. What is the bond angle at the carbonyl carbon of this? And this is also called a beta-lactam. That's nomenclature we learned last Friday. Cyclic amides are called lactams. A four-membered cyclic amide is called a beta-lactam because this is the alpha carbon and the beta carbon. So what is the bond angle at the carbonyl carbon of a beta-lactam? What's that? Yeah, it's basically 90 degrees. What does that bond angle want to be? What's the ideal bond angle? It's 120. It's supposed to be trigonal planar. Okay? So we have a lot of angle strain in this ring. Okay? And that angle strain makes the amide that's part of the beta-lactam significantly more reactive than a normal amide. And that actually is how penicillin kills bacteria. So here we have a slide showing us the mode of action of penicillin. It's going to bind to the active site of an enzyme, a bacterial enzyme that is involved in the biosynthesis of the bacterial cell wall. And it's going to use its other functional groups. All of those other functional groups present on penicillin will allow it to make stabilizing non-covalent interactions with the active site of the enzyme. So once it gets bound to the active site of that bacterial enzyme, there's going to be an alcohol from that enzyme in close proximity to that strained amide carbonyl. So the alcohol attacks, we get our tetrahedral intermediate. The best leaving group on that tetrahedral intermediate is the OH that just attacked. But in this case, if we eliminate out the nitrogen, we release that ring strain that was present. Okay, and that's actually a stabilizing effect. And so that's what happens. This tetrahedral intermediate collapses with elimination of the nitrogen, opening the beta-lactam ring, generating an ester. Usually we don't form esters from amides, but in this case we can do it because the release of that ring strain or that angle strain provides a thermodynamic driving force. Okay? And so now we've inactivated the enzyme. We've covalently attached penicillin to the active site of the enzyme. So the enzyme can no longer produce the bacterial cell wall, and that causes the bacteria to die. Humans don't have this enzyme. Penicillin doesn't bind to the active site of our enzymes in the same way, and so that's how it kills bacteria without killing us. Okay, any questions? Yes? What's that? What does the resistant bacteria look like? Oh, that's a great question. So, of course, there's resistance to penicillin. those resistant bacteria have enzymes that will intercept penicillin and open up that four-membered ring before it makes it to the active site of the cell wall. It has enzymes called beta-lactam-aces that are designed to open up that beta-lactam before it can target the cell wall producing enzyme. Okay? The discovery of penicillin, the story of its discovery is, in my opinion, the most interesting and greatest scientific story of all time. So I'm going to try to save time to tell you that story at the end of class today. I've managed to work that story into at least 2 sacrament meeting talks. There's a lot of great lessons in that story. So hopefully we'll have time at the end of class to learn about the discovery of penicillin. Some of you may know at least parts of the story because it is well known, but you probably don't know the whole story as I'm going to tell it. So to summarize, here we have a table showing us the nucleophilic acyl substitutions that we have learned. And as with all the tables, you're probably getting tired of me saying this, but please don't memorize the table. You should understand why the reactions with the check marks occur, why the reactions with the X's do not. It should make sense based on our knowledge of the relative reactivity of carboxylic acid derivatives. And we've also learned that carboxylic acids themselves are special. We are able to convert carboxylic acids into each of the 4 types of derivatives. And we can also convert all of those 4 types of derivatives into carboxylic acids, in some cases using acid-base chemistry to drive reactions through unfavorable equilibria. Okay, so if you can look at this table and understand why the reactions that occur actually take place and why the other reactions are prevented from taking place, you have a good understanding of nucleophilic acyl substitution. So now we're going to talk about the carboxylic acid derivatives that are used primarily in nature to perform nucleophilic acyl substitutions. There are 2 of them. The first one we'll look at is the acyl phosphate. So here's an acyl phosphate on the right. An acyl phosphate looks a lot like an anhydride. If you turned this phosphorus into a carbon and you didn't have those oxygens, you had another R group there, it would be an anhydride. So acyl phosphates are comparable in reactivity to anhydrides, but they're actually a bit less reactive, and that's good, because if we had acid chlorides or anhydrides floating around in our bloodstream, we would have nucleophilic acyl substitution occurring everywhere in an uncontrolled fashion. And that's not what we want, right? Organic reactions in biochemistry are very tightly controlled. We only want certain reactions to happen, and we only want them to happen at certain times. And so we want carboxylic acid derivatives that are reactive enough to participate in nucleophilic acyl substitution with the catalysis of an enzyme inside the active site of an enzyme, but we don't want that reaction to occur without the enzyme, okay? Because that's how our body can control which reactions occur and which ones don't. So in nature, we use carboxylic acid derivatives that are a bit less reactive. If we wanted to make esters or amides in a lab, we would use either an anhydride or an acid chloride. But in our bodies, we want something less reactive. And so the acyl phosphate is one of those things. So acyl phosphates are made from carboxylates and ATP. We've abbreviated the structure of adenosine here, but this is one of those heterocyclic nitrogen bases that you see in nucleotides. What happens is the carboxylate, remember at physiological pH, most of your carboxylic acids will be deprotonated. You'll have a lot more of the conjugate base than you will of the carboxylic acid itself. So that carboxylate is going to attack one of the phosphorus on the end of ATP. You'll notice we have a magnesium cation here. That magnesium cation serves as a Lewis acid. It forms a Lewis acid-Lewis base complex with these negatively charged oxygens. The effect of that complex is to withdraw electrons from that phosphorus and make that phosphorus more electrophilic. So we attack the phosphorus. We end up with something that looks like a tetrahedral intermediate on steroids because it has 5 groups bonded to the phosphorus. That's okay because phosphorus is a third row element and is allowed to exceed the octet. But it behaves like a tetrahedral intermediate in that it eliminates out the leaving group, forming a phosphorus-oxygen double bond in this case, giving us our acyl phosphate. Okay? Any questions about this mechanism? So another thing I'll tell you is that sometimes when you make acyl phosphates, you do it by attacking the terminal phosphate group of ATP. Other times, you attack the internal group, and when you attack the internal phosphate, this one, you retain the adenosine in your product. You're going to have an adenosine hanging off of here. You never attack the one in the middle. It's always either the one on the end or the one that's bonded to the adenosine, but the mechanism is the same. And if we go to the next slide, we'll see the formation of a fatty acyl adenosine monophosphate. That's what AMP stands for. This is one of those cases where the nucleophile attacked this phosphorus, the magnesium coordinated here, giving us this, and the diphosphate is the leaving group. Right? So sometimes the body wants to use the phosphate without the adenosine. Other times it needs the adenosine on there either for increased water solubility or to be able to make non-covalent interactions with the active site of the enzyme. And so the enzyme that's catalyzing this reaction would put the magnesium in the right place. It would either put the magnesium here or put the magnesium here. That's how you would control that. Any questions on our acyl phosphate biosynthesis? Okay, so the reactions of acyl phosphates, we've got a couple of them shown here. We can make amides from acyl phosphates. Here's one, glutamyl phosphate. This is made from glutamic acid. And then we can react ammonia in the presence of the enzyme. Notice that we use magnesium not only to make our acyl phosphates, but also to activate our acyl phosphates for subsequent nucleophilic acyl substitution. So this magnesium is going to coordinate to these 2 oxygens, Lewis acid-Lewis base complex, making the carbonyl carbon more electrophilic and accelerating the attack of the nitrogen at that position. Okay? It's comparable to the acid-promoted reactions we did, where we would protonate the carbonyl oxygen first and have the nucleophile attack second. Here, instead of using a Bronsted acid, we're using a Lewis acid. We're coordinating the carbonyl oxygen to the magnesium, and that's accelerating the attack of the amine. And that gives us glutamine. So that's how the body makes the amino acid glutamine. It makes it from glutamic acid, first turning that side chain carboxylate into an acyl phosphate, and then having the ammonia attack to generate the product. Questions? And then the next one we see is production of a thioester. Thioesters are also somewhat reactive carboxylic acid derivatives, but less reactive than acyl phosphates. Our bodies use thioesters to produce esters or amides. And the thioesters themselves are prepared from acyl phosphates. So here we have coenzyme A. You've probably heard of coenzyme A. You've probably seen it abbreviated like this. You probably haven't seen it like this very frequently because it is this mammoth massive structure. It is important to know that it's a mammoth massive structure. You don't need to memorize the structure, but to recognize that it has lots of functional groups endowing the molecule with great water solubility and also giving it plenty of opportunities to undergo hydrogen bonding, to form salt bridges and things like that, to be able to be held at a certain place in the active site of an enzyme. So your sulfur can act as the nucleophile, displacing our leaving group right here, giving us a fatty acyl coenzyme A derivative. Again, magnesium is used in this process. And these are key building blocks in biosynthesis. We'll actually talk a little bit about the biosynthesis of select important biomolecules later on this semester, and we'll talk about how fatty acyl CoA derivatives are used to biosynthesize organic molecules. We've got the mechanism of this reaction shown here. And I've already described it generally with the magnesium coordinating to these two oxygens, making the carbonyl group more electrophilic. One thing you notice from this mechanism is the authors of our textbook have done something that I've strongly advised you against doing, and that is combining steps. So you'll see what they've done here is they've done a proton transfer at the same time as a nucleophilic attack. They're deprotonating the thiol of coenzyme A at the same time as that thiol is attacking the carbonyl. So if you were going to do this in solution, it would require 3 molecules to collide simultaneously. You would need the thiol, the base, and your acyl phosphate to all collide simultaneously. That's highly unlikely in a flask, in a reaction that you're performing in a flask. On the other hand, in the confines of an enzyme, of the active site of an enzyme, that can actually happen. Your enzyme active site can actually have a base attached to it, and that base can be in very close proximity to your thiol and to your acyl phosphate. Okay, so that's what active sites of enzymes do. They hold reactants in close proximity. So you can combine these steps only when you're drawing the mechanism of enzyme catalyzed reactions that occur in biochemistry. Okay, so this is legitimate in this case. We can deprotonate the thiol at the same time as it's attacking our carbonyl carbon. We have a tetrahedral intermediate here, and you'll notice there's another combination of steps here. If we were doing this in a flask, we would eliminate out the leaving group, and then we would do the proton transfer after eliminating out the leaving group. But here you can see we're eliminating the leaving group and we're protonating that oxygen simultaneously to get our hydrogen phosphate and our thioester. Okay, so you can combine steps only in enzyme catalyzed reactions. Questions? So this is how our body makes thioesters. What does our body do with thioesters? Well, it converts them into, as I mentioned, usually amides or esters. The nucleophiles that they react with are usually either amines or alcohols in the body. And this is acetyl coenzyme A. Acetyl coenzyme A is the body's version of acetyl chloride or acetic anhydride. When the body wants to transfer an acetyl group somewhere, it uses this reagent as opposed to the 2-reactive acetyl chloride or acetic anhydride. So an example of that is shown here, making an ester from cholesterol. So cholesterol, of course, is a very nonpolar molecule by design because its role is to insert into cell membranes and to impart the correct amount of rigidity to our cell membranes. So cholesterol is not water soluble, but it has to be transported through the bloodstream to get to the various sites in the body so that it can be incorporated into the cell membranes of all of our tissues. And it can't just transport through the bloodstream. So what it does is it gets packaged in these micelle-like structures. This is called low-density lipoprotein, or LDL, where you have phospholipids forming a micelle-type structure. And then you have these cholesterol esters inside. For reasons I don't actually know, to be honest, the cholesterol esters more readily assemble into these micelles than the free alcohols would. So our body will esterify cholesterol using a thioester derivative, a coenzyme A thioester derivative of a fatty acid in the active site of an enzyme. And then that ester will assemble into this low-density lipoprotein. And then that protein is soluble in water. It will float through the bloodstream. But if you have too much of it, then it can start to form these plaques. in your bloodstream. So you don't want too much of this LDL floating around in your blood, or you'll start to get plaques in your arteries, constricting the ability of your red blood cells to flow through your blood vessels and causing problems such as heart attacks or in the extreme case, right? So questions about thioesters and their role in biochemistry. All right, so let's talk now about nitriles. So the nitrile is not a carboxylic acid derivative, but it has 3 carbon-nitrogen bonds in the same way that our carboxylic acid derivatives have 3 carbon Z bonds, 2 to an oxygen and 1 to another heteroatom. So you'll see that nitriles will engage in comparable reactions to carboxylic acid derivatives. First, we need to discuss how we would synthesize a nitrile. Okay? If you were asked to synthesize a nitrile, what would you do? What reactants would you react together to make a nitrile? And I'll give you a hint. You might be asked to do this pretty soon. So it would behoove you to know how to make a nitrile. Yes? Okay, so you can react an aldehyde or a ketone with sodium cyanide and HCl. That's going to make a cyanohydrin. Okay, so that's a specific type of nitrile. What would you do if you wanted a nitrile that was not a cyanohydrin, one that didn't have the OH group? Think back to, yes, Trent. Okay, yeah, I was going to say think back to chapter 7, but Trent figured it out. We can use an alkyl halide, as long as it's not a fluoride. So we'll just use a chloride for now. But cyanide is an anion, right? Our anionic nucleophiles are strong nucleophiles, so they'll participate in SN2 reactions. So we can take either a primary or a secondary alkyl halide and react it with cyanide in an SN2 reaction. And that's going to give us a nitrile. Okay? So 2 ways to do it. If you want a simple nitrile, you're going to use cyanide ion. If you want to make a cyanohydrin, if you want that OH group, then you would use an aldehyde or a ketone with sodium cyanide and HCl. So once you get your nitrile, what do you do with it? Well, there's actually a lot that you can do with it. Unlike one way that it differs from our carboxylic acid derivatives is that it does not have a leaving group attached to this carbon. We've got 3 bonds to nitrogen, but there's no leaving group. So we're not going to see nucleophilic acyl substitution reactions. The reactions we're going to see are more like the ones we saw in chapter 21, and that would be nucleophilic addition reactions. We also saw those in chapter 20. Okay, so we can, because the nitrile has 3 bonds polarized from carbon to nitrogen, we have a strong enough partial positive charge on the carbon that we can have nucleophiles attack that carbon and push the electrons onto the nitrogen. And so that's what's going to happen with our nitriles. So as you'll see, nitriles are versatile. We can convert them into a lot of different functional groups, and that's what makes them very useful in synthesis problems. That's why I said there's a strong possibility you might be asked to make a nitrile at some point in the next 3 days, and because you can convert them into a lot of different types of products. And the first one we'll look at is the hydrolysis of the nitrile, and that affords a carboxylic acid. Okay? So the nitrile is at the same oxidation state as a carboxylic acid. It has 3 carbon heteroatom bonds. So we don't need to oxidize it to make a carboxylic acid. We just need to convert those carbon-nitrogen bonds into carbon-oxygen bonds, and that's a hydrolysis process. We do need either acid or base present to do that. If we conduct the reaction in acid, the product will be a carboxylic acid. If we conduct the reaction in base, the product will be the conjugate base or the carboxylate. We'll go over the mechanism on the screen in a minute, but a key intermediate I'll show you in this process is a species known as an imidic acid. This is called an imidic acid. It looks like a carboxylic acid. If we replace this NH with an oxygen, it would be a carboxylic acid. But because it has an NH instead of an oxygen here, it's kind of like an imine, so that's why we call it an imidic acid. Imidic acids are not that stable in the presence of base or acid. They undergo a highly favorable tautomerization to generate an amide. Okay? And we'll go over the mechanism of that. We first learned about tautomerization back in chapter 11 when we saw enols tautomerize to give ketones. If we replace this NH with a CH2, then that's exactly what we would have, an enol tautomerizing to a ketone. So the mechanism of this tautomerization is going to be the same as that chapter 11 tautomerization we learned. So let's take a look here on the screen at the mechanism of basic nitrile hydrolysis. And as we go through this, we'll just talk about how the steps would be different if we were using acid. You should be familiar enough with these mechanisms by now that you should be able to recognize how it would differ if we were using acid. So step one, if we're using base, then our hydroxide is a strong nucleophile. That's going to attack our nitrile carbon, and that's going to give us this nitrogen anion species, which then gets protonated by water, because we have hydroxide and water present, and that's how we get our imidic acid. So just 2 steps generates our imidic acid species. So how would this differ if we were using acid? If we had H3O plus instead of OH minus, what would the first step be? Protonation of the nitrogen. Yes, so you would protonate the nitrogen with the H3O plus. Then what would happen next? What would our nucleophile be? The nucleophile would be water. So if we were in acid, we wouldn't have OH minus. So protonate first, attack second with our weaker nucleophile. And then because that nucleophile is neutral, we would have to add a third step. We would have to deprotonate that using another equivalent of water, regenerating our H3O plus. So in acid, there would be 3 steps: protonate the nitrogen, attack with water, proton transfer. Questions? All right, so either way, we get to our imidic acid. And so now we have the tautomerization of the imidic acid to the amide. We'll show it in base. We'll discuss how it would happen in acid. So in base, we're going to deprotonate first. So we deprotonate the OH. That generates a resonance stabilized anion. Then we protonate the nitrogen, giving us our amide. How would that differ if we were using acid? What would happen first? There's 2 choices. Yes, Trent? Yep. So we would reverse the order of these proton transfers. You'd protonate the molecule first before deprotonating it if you were using acid. And so we'd protonate on the nitrogen. And you should write this out on your own after class, or now if you're fast enough. Protonate on the nitrogen with H3O plus. That's going to generate a resonance stabilized cation if you protonate the nitrogen. Okay. And then what happens second? It's the thing that happens first in base, which is deprotonate the oxygen. But would we use hydroxide, or what base would we use if we were in acid? We would use water as our base, exactly. So in acid, we use H3O plus as the acid, we use water as the base. In base, we use hydroxide as the base, water as the acid. Okay? Any questions on how we make it to the amide? All right, we're not done yet. This is a three-part mechanism. Once we make it to the amide, if we have water and hydroxide present, we know it's going to happen because it's drawn on the board here right under the screen. We are going to hydrolyze that amide to get a carboxylate. I don't know why they said several steps. It's only 3. I don't really, several to me is more than 3. So I don't know why they scare you by saying there's several steps here, but there's not. It's just these 3 steps we have on the board here, where our hydroxide attacks the carbonyl. We have our unfavorable equilibrium step, and then we follow that by the irreversible deprotonation. Okay, so that's what happens when we hydrolyze an amide in base. If we have acid, then it's the reaction we learned at the end of class on Wednesday, which is comparable to the ester hydrolysis, the reverse of the Fischer esterification, just with a difference in the final step. Okay? So you should be able to draw this out either in acid or base, because we've learned it either at the beginning of class today or at the end of class on Wednesday in the case of the acid-promoted reaction. Okay? So it's a very lengthy mechanism when you consider it all together. So I find it more helpful to break it up into the 3 parts and learn the 3 parts. And it's really only one that's new, and that's this right here, because this is simply the chapter 11 tautomerization mechanism, just with an NH here instead of a CH2. And then this we've just barely learned. So really only this first part to make the amidic acid is the new part. Okay? Questions? Okay, let's talk about what else we can do with nitriles other than hydrolyzing them. So we're able to reduce nitriles, and the outcome will depend on the nature of the reducing agent that we use. And we saw that with carboxylic acid derivatives as well. If we react a nitrile with our strongest reducing agent, lithium aluminum hydride, and then we add water, we're going to generate a primary amine. Okay? We're technically going to be using 2 equivalents of lithium aluminum hydride. We don't specify that because the ALH3 byproduct that is formed also can react. So we don't usually write numbers of equivalents of lithium aluminum hydride, but we just assume there's always enough presence to fully reduce our compounds. So in this case, we would get a primary amine. If we react a nitrile with diisobutyl aluminum hydride, or Dibal, the outcome is different. And then we're going to add acid and water. The book just puts water. You do need acid in there as well, and we'll explain why. What you get in this case is an aldehyde. And this is a reduction because we went from 3 carbon-nitrogen bonds to 2 carbon-oxygen bonds. Okay? So we're going to look at the mechanism on the screen here for both of these reactions. So our lithium aluminum hydride, very strong nucleophile. We know that negative charge is spread out on the hydrogen atoms, not on the aluminum as it is drawn. So we use the aluminum hydrogen sigma bonds as our nucleophile to attack our nitrile carbon. And that gives us this very basic nitrogen anion. that is then going to form a Lewis acid Lewis base complex with the ALH3 byproduct, giving us this intermediate right here. Okay? Now, here's where it gets a little different. So this is an anion. The anion is drawn on the aluminum, but it really resides on the atoms bonded to the aluminum. That anion is present on the nitrogen as well as on those 3 hydrogens. Okay? So this species you wouldn't consider very electrophilic because it's negatively charged. But this carbon is still weakly electrophilic because it has 2 bonds to a more polar nitrogen atom. Aluminum hydride is reactive enough, it's a potent enough nucleophile that it can actually attack this weak electrophile. Attacking it at the carbon, they've combined 2 steps here. I think the author of the book had the enzyme reactions on the brain and decided to combine steps here where she should not have, because this is actually 2 steps here. The one they're showing is 3. What's happening is not 3 molecules colliding together, but rather your aluminum hydride attacks here. These electrons go onto the nitrogen, putting an anion on the nitrogen, and then that will coordinate with the ALH3 that gets released from that process to generate this intermediate. So there's really 2 steps here and not 1. Please do not combine these. There's no enzyme involved. All right, so this thing should make us a little bit uncomfortable. It's a dianion. We don't often see dianions in this class, but this is a delocalized dianion. That negative charge is not, or those negative charges are not localized on the aluminum atoms. They're spread out on all the atoms that are bonded to the aluminum atoms. So it does exist. We add water. That water is going to protonate our nitrogen-aluminum bonds. They really should draw arrows for this step as well. What's happening is our nitrogen-aluminum sigma bonds are grabbing protons from water. You could actually do that in 1 step because there's enough water molecules present that you could have 2 water molecules collide with this compound and do those protonations in 1 step, getting us to here. And then ultimately, these are the byproducts we formed. We're not as worried about that. We're most concerned with how we're forming the primary amine. Any questions about this mechanism? Okay, let's look at what happens when we use Dibal, diisobutyl aluminum hydride, instead of lithium aluminum hydride. Dibal is reactive enough to attack the carbon of a nitrile, but not reactive enough to attack the anion that results. So when we use diisobutyl aluminum hydride, we attack the carbon of the imine, generating this intermediate, and then that intermediate just sits there. Okay? And it sits there until we've consumed all of our diisobutyl aluminum hydride. We actually don't have to run this at low temperature because that intermediate would be stable at room temperature. So it's a little different from the reduction of acid chlorides and esters. But then once you've consumed your Dibal and you've produced this intermediate, then you add water, you protonate that nitrogen, and when you protonate that nitrogen, you get an imine. Now, if we have acid present, which we do, it's unfortunately not drawn here, but if we have water and acid, now that water and acid can hydrolyze the imine to form the aldehyde. And that was a mechanism we learned in chapter 21. That does have more than 3 steps, so I guess the word several would apply in this particular case. Questions on Dibal reduction of nitriles? Okay, there's another reaction of nitriles that is very useful. It doesn't involve reduction. It involves the use of a Grignard reagent or an organolithium. I'll show it to you with a Grignard reagent, but it would work with either a Grignard reagent or an organolithium species. Okay? So these are strong nucleophiles. It shouldn't be surprising that these can add to nitriles in the same fashion as our metal hydrides can add to nitriles. Okay? So that's going to give us an anionic intermediate, very similar to the one we see up here on the screen. And then when we add our water, we of course protonate that nitrogen, giving us an imine. And then if we have aqueous acid, you could write it this way. You could write H+, you could write sulfuric acid. You're able to hydrolyze that imine. And what you get is a ketone. So this provides us an alternative way of making ketones via carbon-carbon bond formation. We learned back in chapter 20 that we could do this with acid chlorides and organocuprates. We can also do it with nitriles and Grignard reagents. And when we think about where the nitrile comes from, it allows us, in terms of retrosynthesis, to take a ketone and to chop that ketone up into 3 building blocks. Okay? So our R group over here, that would come from an alkyl halide, because up here we learned that you can react an alkyl halide with cyanide to make a nitrile. Okay? So then the carbonyl group, that's coming from our nitrile, or from our cyanide anion. Okay? So our cyanide anion would react with an alkyl halide to make a nitrile. And then that nitrile intermediate would react with a Grignard reagent. And what do Grignard reagents come from? They come from alkyl halides and magnesium. Okay? So if we take it back to the precursor of the Grignard reagent, we find that we can combine 2 different alkyl halides and cyanide. Of course, you don't just mix these 3 together, but ultimately we can derive a ketone from 2 alkyl halides and cyanide. We do an SN2 reaction, we make a Grignard reagent, and then we react the Grignard reagent with the nitrile to generate a ketone. Questions? So, to summarize, we can convert nitriles into carboxylic acids, primary amines, aldehydes, or ketones. That's 4 different functional groups. So you can see how nitriles are versatile and would be useful in synthesis problems. Okay? All right, we do have about 3 minutes left, so let's talk about penicillin, the discovery of penicillin. The story begins in 1928. There was a researcher, a Scottish doctor in London. His name was Alexander Fleming. He was studying bacteria, streptococci, like strep throat bacteria. And so he had petri dishes of these streptococci in his lab, and he went on vacation. This was summertime, and he left his petri dishes out on the lab bench. When he came back from vacation, he found that there was mold growing in his petri dishes. Okay? What would you do if you were a scientist and you came back and you found your mold was contaminating your petri dishes? What would you do? You'd throw it away and start over. But that's not what Alexander Fleming did. He looked closely at those petri dishes and he saw that the bacteria that were in contact with the mold were dead. So he realized there was something in that mold that was killing bacteria. Now the mold happened to be from a species known as Penicillium. And so as you can connect the dots, you can see that there was penicillin produced by that mold that was killing the bacteria. Now in 1928, they did not have effective antibiotics. They had some early first generation ones that were kind of hit or miss. And so Alexander Fleming recognized the significance of his accidental discovery. And it was truly an accident, because what had happened is he left the windows of his lab open, which was common because they didn't have air conditioning in 1928 in London. There was another researcher 1 or 2 floors below who was studying penicillium. And so the mold spores blew out the window of the one guy's lab and into the window of Fleming's lab and contaminated his Petri dishes. So it was a total accident. But he realized the significance of it, and he immediately shifted the focus of his research to finding and isolating the compound in the penicillium that was killing the bacteria. But it was difficult. Penicillin is not that stable. If you take penicillin, you have to store it refrigerated, right? It degrades at room temperature. It took him 13 years to isolate enough penicillin to start a clinical trial. So in 1941, in conjunction with Oxford, they dosed the first patient with penicillin. The patient's condition improved. They had no idea how much penicillin to give. They didn't give enough. So once the penicillin ran out, the patient died. Right? There wasn't enough penicillin left to give. So they isolated more penicillin, gave the second patient a larger dose. That patient initially got better, but also ended up dying because they didn't isolate enough. It was on the third try that they isolated enough penicillin to actually heal the patient. By now, it was 1943. Okay, between 1941 and 1943, what was happening in England? Think back, history. World War II, the Battle of Britain, they were getting bombed, right? So they were afraid their lab was going to get bombed. It's not an ideal scenario for performing research. So in 1943, Fleming moved to the US. He took his molds, he packed them up in his luggage, he actually smeared some of the mold inside of his coat. He tried to figure out how to get as much mold as possible from England to the US. And he went to Peoria, Illinois. There was a US Department of Agriculture lab in Peoria, Illinois. And the problem they faced was that the penicillium mold he was working with initially was not producing very much of the penicillin. That's why it took him 13 years, and that's why they had to keep going back to the beginning and isolating more and more to treat their patients. So what they did was they put out a call to the citizens of Peoria. They said, bring us your moldy fruits and vegetables, and we will scrape the mold off of them and see if that mold is producing more penicillin than the mold we're working with now. And it turned out they found a mold on some woman's moldy cantaloupe that she brought into their lab that actually produced a lot more penicillin than any of the molds they were using before. So that was a breakthrough. But then they had to scale it up because, as I mentioned, World War II was going on at this time. And if we go back in time to World War I, infections were lethal because there were no antibiotics. And so it was more common for soldiers to die of an infection that they had occurred due to a wound than it was to actually be killed on the battlefield. Right? So you would survive getting shot, but then your wound would get infected and you would die of the infection. So the US pharmaceutical companies scaled up the production of the mold and the harvesting of the penicillin from the mold, much in the same way that you see them now scaling up the production of mRNA vaccines and adenovirus vaccines. So there are some parallels between this story and what we're seeing this year. So they were able to scale up about 100 times in the case of, in the span of a year, the production of penicillin. And so they were able to give it to the Allied soldiers on the battlefield and save probably tens of thousands, if not hundreds of thousands of lives of soldiers due to being able to heal those wounds and prevent the infection of those wounds due to the mechanism that we showed you here on the screen. So the nuclear bomb is usually viewed as the most significant scientific advance from World War II. I would argue that penicillin is at least as significant. It saved a lot of lives in the war. It's saved a lot of lives since the war. The lessons we can learn from the story are twofold. Number one is you have to be persistent. It took him 13 years to make this discovery, right? He didn't give up. And number two, many times scientific discoveries are made by unexpected observations, by being very, very observant. So instead of just tossing out his Petri dishes, Fleming looked closely at them and noticed something he wasn't expecting. All right, we'll stop there. Good luck on the exam. We'll see you on Monday.